Good morning, good afternoon, or good evening, depending on where in the world you're watching from. Thank you for joining us for the third day of Cell and Gene Therapy Digital Week, a live webinar series brought to you by the producers of the face-to-face conference, Cell and Gene Therapy, Bioprocessing, and Commercialization. This year's event takes place in Boston, September 25th to 28th as part of Biotech Week Boston. I'm Eric Warner, Marketing Manager at Connect 365, the world's leading facilitator of knowledge sharing and business connections. We hope you enjoy all the expert-led sessions and resources available to you as a Digital Week attendee. If you like what you see, please click the Invite a Colleague button or click one of the on-screen social media buttons to invite others to join. Before we begin the session on platform processes for PSC-derived product manufacturing, presented by Dr. Nick Timmins, I want to remind you that should you have any questions, please type them into the control panel below the slides, and we'll do our best to answer them during the Q&A session immediately following the presentation. Without further ado, I'll hand it over to Dr. Timmins.
Welcome everyone, my name's Nick Timmins and I'm the Vice President of Technology at CCRM, and this presentation is going to describe the development of a platform process for pluripotent stem cell derived product manufacture. Before I do that, I would like to describe briefly what it is that CCRM does. And so on the left hand side here you can see one of the things we do is build companies, and we do this utilizing a team of business, regulatory and IP experts that have a background in technology commercialization, and that background is more specifically in regenerative medicine and cell and gene therapies. Unlike, for example, a typical university tech transfer office, we actually have our own wet labs, and what this allows us to do is advance the research itself. We do this in a number of ways, and one way is through providing cell reprogramming and gene editing services. We can also take very early stage methods and technologies and translate these up to the next stage. For example, taking a benchtop cell culture process in multi-well plates and moving that across into a small scale bioreactor, and in doing so developing up robust protocols. Going beyond merely advancing the research, we can actually bridge the gap from these preclinical type methods all the way through to commercial ready cell and gene therapy manufacturing processes. So we tackle things such as the process optimization, scale up, scale out, closure, automation and risk reduction. And then onto the right side of this image here you can see that we also deliver products, and that capability will come on stream in 2018 with GMP facility for cell and gene therapy manufacture. And all of these capabilities are available as a contract service. In offering these different capabilities, CCRM is home to what is known as the Center for Advanced Therapeutic Cell Technologies. This is something we're very proud to host, and it's a $40 million collaboration between CCRM, GE Healthcare and the Government of Canada. And it's all about employing the knowledge and skill sets of GE and CCRM to develop the next generation of solutions for the effective manufacture of cell and gene therapy products to make sure that these treatments are more accessible to patients. And at the bottom of the screen there you can see an image of the Prime Minister of Canada, Justin Trudeau, announcing this funding in January last year. So moving along to the meat of this presentation, it's worth asking, why do we need a better process for pluripotent stem cell manufacture? This is the platform process that I refer to in the title presentation. And the reason is the amount of cells that we're ultimately going to need to make in order to deliver our therapeutic products. So this table is outlining some of the cell number requirements for 4 fairly promising therapeutic indications. The top row here, myocardial infarction, the target cell type in this case would be cardiomyocytes. A number of publications have suggested that doses in the range of 10 to the 8 to 10 to the 9 will be required. There's 500 annual new cases of MI in the US alone, and if you multiply those numbers through, you come out with an annual cell requirement of around 2.5 by 10 to the 14 cells. If you do that same sort of exercise for type 1 diabetes, spinal cord injury, and acute on chronic liver disease, you can see you get some very large cell numbers. Now, of course, not all of those cases would be potential recipients for a cell therapy, and of those that are, you're not going to get 100% market penetration. So if I sum up that right-hand column and take just 1% of that number, it still comes to 4.6 by 10 to the 12 cells. If we make a further assumption that each one of those final product cells needs one pluripotent stem cell from which to generate it, and we assume we're in a planar cell culture system where we get 300,000 cells per centimeter squared, that's the equivalent of 600 40-stack cell factories. Those cell factories are big, they're cumbersome, they need mechanical assistance to move them around. They take a lot to work with, and they also don't benefit from any sort of instrumentation or automation. In contrast, if we make the assumption that we use a suspension culture system, and we are hitting a density of around 5 million cells per mil, this is the equivalent of a 900-liter scale culture. And that is something which is much more manageable. So how do we go about developing a process in which we can do a 900-liter scale cell culture? That's outlined here along the top, and this is what we've done at CCRM. So starting with the research methodology for pluripotent stem cells in planar culture, many labs actually do this in 6-well plates, let alone T-flasks. And at CCRM we take that across into a stirred tank reactor format, beginning with the AMBER 15 bioreactor system. So this platform has 24 little tic-tac box size bioreactors in place, and that allows us to do multi-parametric screening of different process parameters. Once we identify some starting points for further work, we translate those cultures into benchtop bioreactor systems in the sort of 100 to several liter scale process. This allows us to do further development in a fully closed and automated and instrumented system. And from there we go up to a production level systems in a single use format, and we can take those eventually all the way up to 2,000 liters. So in this context, what we're actually doing is growing the pluripotent stem cells as aggregates in these stirred tank bioreactors. And we do it as aggregates because it's surface free and we're not adding any particles. If you were to use microcarriers, for example, you have to get those out at the end of the process. And not only do you have to worry about the microcarriers themselves, but you have to worry about any little pieces that may have broken off those microcarriers. And that can be a pain. As I mentioned, these STRs can go all the way up to 2,000 liters in single use, and we can utilize the mixing parameters to manipulate the characteristics of aggregation. So starting with that AMBER 15 system, these are 15 mil cultures, and looking at a variety of different parameters, these are just two examples of what we looked at. So starting at the top, we have the feed profile, where we're looking at no change, a 50% daily medium exchange, or a 100% daily medium exchange. And the graph at the top left there is looking at the cell expansion that was achieved at each day. So this is the cell number on the day that we're doing the measurement divided by the number of cells that was put into the reactor at the beginning of the experiment. And what you can see, unsurprisingly, is that if you don't feed your cells, they don't grow very well. If you give them a 50% exchange, they grow, and if you give them a 100% exchange, they grow a bit better. On the right-hand side there, you can see what happens to the pluripotency profile, and we measure this through the expression of TR160, SSEA4, OCT4, and SOX2. Again, unsurprisingly, if you don't feed these cultures, the pluripotency drops away fairly dramatically. If you feed them at 50%, it's still fairly good, and it's a little bit better if you feed them with a 100% exchange. Looking at the bottom set of graphs, we're now exploring what happens with different inoculation densities. So we're looking here at 200,000 cells per mil versus 500,000 cells per mil. And focusing on the expansion graph again, what we can see is that at 500,000 cells per mil, these cultures seem to plateau out after around 4 or 5 days, whereas those at 200,000 per mil keep on growing. And what's happening here is that the higher cell density, the aggregates are reaching a limiting size at which mass transport limitations set in, and so they just can't grow any further. Focusing on the right-hand chart now for pluripotency, what we see in contrast to the growth is that the pluripotency remains good regardless of the inoculation density. So utilizing the data that we obtained from the AMBER, we picked some initial starting parameters to go into the next stage bioreactor at the 125 mil scale. So this is an 8-fold scale up from the AMBER, and what you can see here is that the fold expansion was pretty comparable, and so was the pluripotency. So we're quite happy with these data. But so far, this data is all for a single passage, and ultimately we need to be able to grow these cells over multiple passages, because as those aggregates get big and stop growing, we need to dissociate them back down to single cells, put them into a fresh culture, and grow them up again to keep increasing the total cell number. So looking at the expression of the pluripotency markers over 7 passages, shown on the left here, you can see that it's actually pretty good. There's a bit of a dip there at passage 2, and this is something we see fairly consistently with this particular cell line, and we believe it's related to the adaptation of these cells to aggregate passage, because they are initially coming off a monolayer culture. On the right-hand side, this is the gene expression following differentiation of these cells after passage 7. You can see that our pluripotency genes OCT4 and NANOG are downregulated, while our differentiation-associated genes have been upregulated. So again, this is nice and what we would like to see. However, I do have a dirty little secret, and that's that the data so far were all generated using the HES2 cell line. And HES2 is a somewhat special cell line in the world of pluripotent stem cells, and it is not a cell line that you would really want to be using if you were going to put your final product into humans. So naturally, we wanted to do some work with a cell line which would be much more suitable for the manufacture of a therapeutic cell product. And what you can see now is data generated doing these cultures using the ESI-17 cell line. So focusing on the top graph first, and starting from the left with this grey line, this culture was first generated using a single vessel, and we grow that up over a period of time, and then we've passaged that culture, which is the vertical dashed line, and split that single vessel across 4 separate vessels, and we've seeded them at slightly different densities. We grow those up again for a while, we've passaged again, grown again, passaged again, and grown again. So what you can see here is that the growth profile is fairly consistent. Across the vessels it looks more or less the same, and the fact that we've inoculated slightly different cell densities is also tracking relatively well. The bottom graph is essentially a repeat, although it's a completely independent repeat, and it was even done in slightly different bioreactors. But once again, we have our initial culture period with a single vessel. It's then been split across multiple vessels. That has been split again. We've grown them up a little bit further, and this time we've taken those aggregates off and sent them into differentiation. And that's something I'll talk about a little bit later in this presentation. So that's all looking good. Once again, we know we can serial passage, and we can do it with our ESI-17 cell line. We need to keep going upwards in scale, and this data is showing you a 130-fold increase in cell culture scale. So starting on the left-hand side, we have a 125 ml culture that was passaged and then brought up to 750 ml. That was passaged again and brought up to 2 litre. And you can see that despite the change in vessel size, the growth profiles remain relatively constant over time. What do those cells look like at those different stages? You can see that here at the top left, where we're looking at the fold expansion, so the 125 ml 1 litre and 2 litre cultures are all coming in around about the same place for their fold expansion. And then off the back end of the 2 litre cultures, you can see on the top right there what the FACS profile looks like, where we've got good expression of SSEA4 and TRA160, and those double positive cells are also double positive for SOX2 and OCT4. Down the bottom here, you can get a feel for what these cultures actually look like. So day 1 there, this is after putting the cells in as a single cell suspension. They spontaneously aggregate to give you these little balls of cells. Day 3, they've gotten a little bit larger in size, and then by day 5, not only they're larger in size, but you can see they're starting to get this sort of dense characteristic to them as the cell mass has increased. Moving up even further in size, we will come to the next level of bioreactor that we've chosen to use, which in this case is GE's Accelerix XDR10 system. This is a single-use stirred tank bioreactor with a maximum operating volume of 10 litres. It's fully instrumented and controlled, and the product family scales all the way to 2,000 litres. And you can see on the bottom right there, this is what the 2,000 litre system looks like. So moving up to that next level and achieving an over 500-fold scale up relative to where we started in the amber, this is what happens. On the left-hand side of this graph here, we've started in a 100 ml bioreactor, and then we've gone through a series of several bioreactors to get it up to the 10 litre XDR and brought that up to an operating volume of 8.2 litres, giving us around about 8 by 10 to the 9 cells in total. So just walking through this graph a little bit more closely, the blue line starting on the left, this is the 100 ml bioreactor. We've passaged that and dropped it into the next stage bioreactor, which is shown in the orange line. That is then passaged and put into the next bioreactor, shown in the grey line, and then the yellowy orange line there at the end is the 10 litre XDR system. The coloured bars that are going upwards there at each of the passage points, these reflect the pluripotency as measured by those 4 markers, TRA160, SSEA4, OCT4 and SOX2. You can see it all looks pretty good. At the first passage there it was a little bit below our target of 85%, but it recovers quite nicely and we keep it above that target range thereafter. So just to reiterate, this was done using the ESI 17 line. It was done in M-teaser media. The cells were single cell passage at the beginning of the process and then for each of the subsequent passages utilizing ROCK inhibitor. And we were very, very happy with the outcome of this experiment because this was actually the first time we'd grown these cells in the XDR10 at all, and it effectively worked right off the bat. So that's great. We can make lots and lots and lots of pluripotent stem cells. The problem is that the pluripotent stem cells themselves aren't really all that much use. You wouldn't want to give these to a person unless you don't particularly like that person because they're probably going to cause problems. What we're really interested in is the differentiated progeny of these pluripotent stem cells and using those as our therapeutic product. And something that's been a bit of a focus area at CCRM over the past couple of years is the generation of cardiomyocytes. So why would you want to make cardiomyocytes from pluripotent stem cells? Well, maybe you could use them to replace or restore damaged heart tissue. Heart failure outranks cancer in total life years lost, and it cost the US $31 billion in 2012, and that's projected to reach $70 billion in 2030. And there's good proof of concept data out there that this approach might actually work. So what you can see down the bottom here comes from an article published in Nature in 2014 from Chong et al. And the green area is human cardiomyocytes derived from pluripotent stem cells, which have been injected into the infarct region of a monkey's heart. And that infarct region is delineated by this dashed white line. You can see the cells are sitting there and they've been grafted into that environment. Just like the PSC of themselves, of course, we're going to need to make lots and lots of these cells. How do we go about doing that? Well, the approach we've taken is effectively the same approach that we took for scaling up the pluripotent stem cells. We started off with a method quite literally in 6-well plates, translated that to the amber and developed some understanding of what's happening in our process, moved that to the benchtop bioreactors, and will be in the future taking that to the larger scale production systems. The differentiation protocol that we were working with here is outlined down the bottom, and it started from monolayer pluripotent stem cells, which are disaggregated to single cells and then placed into low attachment 6-well plates. There they were left to spontaneously aggregate for a day, at which point the media was washed out and replaced with a medium that was contained BMP4, BFGF and activin A. After 2 days, that media was completely washed out and replaced with a fresh media that contained a Wnt inhibitor. That was left to grow for 4 days, at which point fresh media was exchanged with no further modulators, and that was carried on for every 2 days thereafter. I'm showing here taking this process out for 12 days. It doesn't need to be 12 days, it can be longer. That's really up to the person's choice who's doing the work, depending on what they're trying to achieve down at the end. Now, up front there we had this process where we were taking monolayer cells and disaggregating them and forming aggregates from them. Well, in our case, we already have aggregates of pluripotent stem cells because we've just generated them in our bioreactor systems. So we front-end this process directly on to the differentiation process. In doing this, one of the challenging aspects in terms of the differentiation method itself is understanding exactly what's happening with the induction of differentiation and the inhibition of Wnt signaling. And so this was something that we wanted to explore, exactly how these two elements interplayed with each other, and to do so we utilized the AMBER 15 bioreactor system to look at how these effects interact. So what you can see now on these graphs here is an exploration of how the timing of Wnt inhibition, expressed as time after the differentiation has been induced, interacts with the concentration of the factors BMP4 and activin A. So the left-hand graph here is showing you what the impact of those two factors is on the percentage of the cardiac troponin T positive cells that you'll get out at the end of the culture. So those CT&T positive cells are what we are defining as our cardiomyocytes. And then on the right-hand graph, you're looking at the impact of these factors on the total cell yield. So focusing on the CT&T positive cells first, what this data is telling you is that the harder we hit them with the growth factors and the earlier we inhibit Wnt signaling, the more CT&T positive cells we get as a percentage of the total cell population. In contrast, however, when we look at the total cell population, or the total cell yield, the concentration of growth factors doesn't really make any difference at all, and the later we inhibit Wnt, the more cells we get at the end of the day. And so we have two effects, both of which we're interested in, but they're working in slightly opposite directions. And this is important because what we're ultimately interested in is the final cell yield, but of the cell type that we're interested in. So ideally, we would like to maximize the expansion and the differentiation efficiency. But as our data was showing us, in this particular case, the two are working against each other, so we have to come to a point in the middle somewhere that is going to meet all of our needs. So there is a caveat here and a point I'd like to stress for consideration. So the caveat is, what's the interplay between differentiation efficiency and the purity of your final product, and what is acceptable in terms of purity for your final product? If it's not highly pure, what are the other cells that are in there? Are they benign? Are they helpful or beneficial in some way, or are they detrimental? And if they're detrimental, for sure, you want to make sure that you can either get rid of them or your differentiation efficiency up front is very, very high. You also need to consider where is the expansion most effective versus where is the differentiation most effective. And what I'm referring to here is, say you have a situation where you do need to get a very, very pure end cell population, and you're going to try and achieve this by maximizing your differentiation efficiency. This is going to result in a very low expansion during differentiation, and in fact, if you push it too hard, you're probably actually going to lose cells. However, we have this wonderful process up the front for generating very large quantities of pluripotent stem cells. So it may actually be more cost-effective to generate more cells up front, knowing you're going to suffer losses in differentiation, but then not have to go through complex downstream processing in order to remove any contaminating cells. So transitioning back to a look at what's happening in our bioreactor systems, this is comparing 4 different runs of cardiomyocyte differentiation from PSC, and in the black bars you can see the percent CT&T positive cells, and in the orange bars you can see the yield. So there's a little bit of variability here, but each one of these 4 runs is coming in at a cardiomyocyte yield of over 5, which is quite nice. And then down the bottom there you can see what the composition of the cell product from each of these runs was. So they're predominantly ventricular cardiomyocytes. There are some atrial and nodal cells in there, and then there's some non-cardiomyocyte cells in there. And these are predominantly believed to be cardiac fibroblasts. At the top right, you can see an image of what one of these cardiomyocytes looks like. CT&T is shown in green, myosin light chain 2 shown in shown in red, and DAPI is blue and is of course our nuclei. All of that data, again, that I've shown you has a secret to it, and that was that it was generated with HES2 again. So as we did with the PSC expansion, we also need to demonstrate that this works for a more clinically relevant cell line. So the data now shown at bottom right is for ESI 17, and in this case you can see over a number of ones we get a mean percent CTNT positive cells of 67 and the yield of CTNT positive cells of 2. So you'll see there in these data are somewhat variable and they're not as strong as the HES2, but this was some very initial data before we'd done any real process optimization for this particular cell line. That's something we're doing now, and so far we've been reasonably successful, but I like to present this particular data set because it shows how different cell lines behave a little bit differently, and it's also a nice complete data set to be presenting as well. So our cells grow well, they look like they're cardiomyocytes. Thankfully, they also behave like they're cardiomyocytes. So what we have at the top of this slide is a look at the electrophysiology. As you would hope, if we take a ventricular-like cell, we get a ventricular-like waveform. Similarly, for the atrial cells, we get an atrial-like waveform, and our nodal cells have a nodal-like waveform. The set of graphs shown at the bottom half of the slide there are illustrating what happens if you expose these cells to cardioactive drugs. Along the top we have the field potential difference, and along the bottom we have the change in beat period. And I'll speak more to the beat period because this is a little easier to grasp conceptually. So this is quite literally the change in time from one beat to the next. So on the left-hand side there, this is looking at what happens if we add isoproterenol. And the more isoproterenol we add, the smaller the beat period, which means the cells are beating faster and faster. And this is exactly what we would anticipate happening. So all of that data, not only was some of it largely HES2, but it was also done in the smaller scale bioreactors going up to as much as 2 liters, but we also wanted to demonstrate this at the 10-liter scale, taking our PSC that we generated in the XDR10. So what we have done here is we grew those PSC up, the ESI 17 and the 10-liter reactor. We then took that material as the aggregates and we split it into 2 different vessels for cardiomyocyte differentiation. And that's what you can see on those flat plots there as A and B. And so for each one of those you can see that the CTNT positive cells come in at around 97%. So differentiation efficiency has been very, very good there. The right-hand panel there you can see the cells twitching. This is the cardiomyocyte spheres beating away in culture. And the yield of cardiomyocytes per input pluripotent stem cells in this culture was greater than 5. To sum things up, we were able to successfully transition pluripotent stem cell expansion and differentiation to stirred tank reactors. We demonstrated scale-up over several orders of magnitude. Our process represents a pathway to further scale-up in single-use systems. The approach enables process closure and automation, and we've demonstrated this for multiple pluripotent stem cell lines. So I showed data from 2 of them here, and we're actively working on additional cell lines at the moment. And the other thing we're looking at is additional differentiation lineages as well. And finally, I would like to make a number of acknowledgements. Of course, the CCRM technical teams who have so diligently worked away at this process. The list of names now would be quite long, so I would broadly acknowledge the team and their work here. The electrophysiology work was done at the University of Göttingen by Drs. Glauben-Schmidt and Dowski. The cardiomyocyte differentiation protocol came to us from the McEwen Center for Regenerative Medicine and Gordon Keller's lab there. We've received support from UHN's Peter Monk Cardiac Center. The work in the AMBER system was supported by Sartorius Stedham. And of course, I would like to acknowledge the Federal Government of Canada and GE Healthcare for their involvement, not only as a funding partner, but GE has been directly involved in some of the process development activities for the pluripotent stem cell expansion. And with that, I would thank you all very much for your attention.
Excellent session, Dr. Timmins. Looks like we have a few questions from the audience. first question. How did you passage the cells?
Yeah, so there's a few different ways to do that depending on what scale one is working at. But once we get up to the larger scales there, we broadly take either an enzymatic approach, similar to what you might do in a flask using enzymes such as triple. And we also have non-enzymatic approaches where one can use physical breakup of those aggregates and move them across into the next stage. When we do an enzymatic passage, we take those all the way down to single cells and we put them in as a single cell suspension into the following bioreactor.
Great. Next question from the audience. How are media exchanges or feeding achieved?
So again, it really depends on what scale one is operating at, but focusing on the larger scales, you can use several different technologies. Examples include TFF filtration modules, and something that we've been working a lot with here at CCRM is acoustic perfusion systems, where we basically use acoustic waves to trap the aggregates and prevent them from leaving the system when we're pumping media out. And as that media is going out, we're pumping fresh media into the system simultaneously. And you can also use various combinations of different approaches as well. In the smaller scale systems, such as the amber, you can't use these perfusion technologies in the same way. So we actually pause the stirring on the system. We allow those aggregates to settle out briefly, and then we go in and remove the media, leaving the aggregates on the bottom, and just add the fresh stuff on the top.
All right, down to the last question from the audience, I believe. How big do the PSC aggregates get?
We aim for around 300 microns, and you can wait longer, and of course they will get bigger as well. So they will grow up to 400 to 500 microns. Sometimes they'll go even bigger than that. But at that stage, they start to spontaneously differentiate, and in the center of those aggregates, you even start to see a degree of senescence. So by keeping ours in that sort of 300 to 400 range, we have good pluripotency throughout, and we are able to maintain nice robust expansion over the full course of multiple passages.
All right, that's it for questions. Thank you, Dr. Timmins, for the presentation, and thank you to the audience for watching. As we're wrapping up this session, just want to remind everyone that all the sessions will be available on demand within the next 48 hours. Right now, you can download the slides from this presentation as well as a wide selection of white papers and videos by clicking the Resources tab on the top of your screen. We'll see you tomorrow for 2 sessions, Building a Sustainable Academic Engine for Feeding De-Risked Assets into the Biopharma Pipeline by Dr. David DeGusto, and Making Cell Therapy Process Changes by Greg Rosati. And that's it for now. Have a good day.